Hey guys, so we are going to start looking at Unit 2, and this is the first video starting with Chapter 9, the cell cycle. We're going to try and fit this into 2 videos, so hopefully this won't be a long chapter. Okay, so let's jump straight into it. So this is the first video, and we're going to look at an overview of the cell cycle in this video, and then looking at mitosis. Okay. Before we actually get into it though, I just want to show you guys something pretty interesting. This is a image graph of the development stages in the human embryo. Yeah, so it starts here in stage 1, which is when the zygote forms. Now the zygote forms when the egg and the sperm join together and forms the first cell of a new individual, yeah, and that's called the zygote. And then it progresses through a series of different stages in early development to sort of show sort of how these how the baby develops. A typical pregnancy lasts about 9 months or 40 weeks, and this shows sort of up to about 60 days or 2 months. Okay, so I just want to point out a few things. Yep. So the first thing to note is around about week 3, you've got at about stage 10 here, the embryo actually develops a heart. At about 30 days, stage 12, it develops a brain. It develops eyes, ears, kidneys, livers, and the heart just begins to pump at around here. Okay. At about week 6, so around about 14, 15, you have movement. You've got hands and feet, and then week 7, you've got movement and brain function. Okay, by week 8, the body is essentially complete, and it now progresses from an embryo into what we call a fetus. Yeah, in which case most of the major organs have finished developing by this stage, and it really is just growing in size. Okay, by about week 10, they hypothesize that the baby already has consciousness, and by about week 20. It's already entering into a rhythm of waking and sleeping, and at about 28 weeks, it is pretty much ready to be born. Yeah, at 28 weeks, which is kind of like the earliest stage in which a premature baby can be delivered. Now, one of the interesting notes is that in Victoria, the legal sort of say number of weeks, or the earliest then you can abort a baby is actually at 24 weeks. Which is quite surprising considering, from what we understand about embryonic development, majority of the major organs have already finished early on, and the baby is essentially complete way before then. So one of the key points I guess I just want to bring home is that some of the reasons why we are learning this is to understand how science plays an important role in the decision making that we make in our society. Yeah, and I just want to sort of illustrate the point that to me, life starts at conception, not at birth. Okay, and so I just wanted to point that out to you guys and let you guys sort of be informed in some of your understanding of these concepts. And as we sort of progress throughout this unit, you're also going to be able to tackle some of those ethical issues as well. Okay, all right, let's jump into the cell cycle now. For the majority of cells, cell reproduction is when a cell or a organism creates a new generation of single cells. So this is a broadened definition from organism reproduction, and basically it kind of highlights how cells reproduce for an organism to grow. Yeah, so when you and I grow, our cells are not necessarily becoming bigger. In fact, growth happens through cell division. Yeah, the cell divides, and you actually just have more cells in the millions that are identical, that then form your major organs, major tissues, and stuff like that. Okay, and so here's just a bit of a diagram of that embryonic development. And you can sort of see there you've got one cell in day 1, and then in day 2 that one cell divides into 2, 2 divides into 4, 4 divides into 8, and then it progresses as it goes along until you have millions and millions of cells that form your organism. Okay, now the reason why cells divide is first and foremost for growth and development. Yeah, so an organism grows when the cells divide and specialize into different organs, different tissues, and things like that. But also, cells need to divide in order to replace the other cells that have been damaged, that are worn out, have sort of lived their lifespan, or are perhaps infected or diseased, and so they need to be replaced. A good example of that being scar tissue and things like that. Okay, this table sort of shows the typical lifespan of a cell, and you can sort of see when the cell reaches the end of its lifespan, it needs to be replaced. Yeah, and so intestinal lining. So when you are digesting your food, the cells that line your gut, particularly your intestines, only last about 1.3 or so days. Yeah, your stomach lining about 3 days, your tongue about 3.5, cervix about 5.7, and as you sort of go down your cheek, about 10 days. The air sacs in your lungs will last about 21 days. Your red blood cells will last 120 days, or roughly give or take 4 months, give or take a couple of days there. And then kidneys 170, and you can sort of see right down the bottom, your nerve cells are the ones that tend to live the longest, typically spanning your entire lifetime, because they are the ones that form kind of your brain function, memories, and things like that. Okay. Now, in order for cells to divide, two things need to sort of occur. The parent cell, which is the original cell, needs to pass on to the cells that have replicated the necessary genetic information. That is the DNA that is found in the nucleus. It then also needs to pass on the organelles onto the daughter cells as well. And each of these sort of have a different name. When the cell replicates the genetic material and divides the nucleus, we call that process mitosis. Now, this is a very important term because we are going to be focusing on it for the majority of this chapter. But mitosis is the replication of genetic material in the cell, or otherwise known as the division of the nucleus of the cell, because the nucleus is the organelle that holds all of the DNA. Okay. Now, the other half is what we call cytokinesis. Cytokinesis is when the rest of the cell begins to divide. And so you can sort of see there this kind of contrast between the nucleus dividing and then the rest of the cell splitting up and dividing as well. And you'll sort of see why, as we unpack why these two processes are really important. Okay. Here is just an overview of this cell cycle. Now, it's important to remember that this cycle sort of happens constantly. Yeah, so whenever a cell divides, it sort of enters into this cycle, and there's sort of say no start or end phase. But for the sake of simplicity, we're going to start right about here and work our way around. Okay, so we're going to start with what we call the interphase. This particular 3 stages here are all called interphase. The word inter meaning in between, and so interphase is between the cell division. So this is during the life of a cell when it is not dividing, and that's broken up into 3 particular phases. Okay, the first one is called the G1 phase, or the gap 1 phase, which is particularly marked by cell growth. A few other things also occur during this time, but the main thing is that the cell is growing. The S phase, which is the synthesis phase, and during this phase the DNA is replicating. Okay, and then the G2 phase is the second gap phase, in which the cell is then preparing for mitosis, and then from there it now exits interphase and enters into mitosis, or end phase. Yeah. Now, mitosis is part of cell division. Okay, and it's encompassed by 4 stages. The first one is called prophase. The second one is called metaphase. Anaphase is the third one, and the last one is called telophase. Now, we are going to be discussing each of these in details later on in this video, but this is just an overview so that you guys can see the 4 different phases of mitosis. Yeah, the acronym that it makes is PMAT, and it's just an easier way to remember it. And then after mitosis occurs, cytokinesis also then occurs, which is the division of the rest of the cell. Okay. Now, all of these particular section here Prophase, Metaphase, Anaphase, Telophase, and cytokinesis makes up the cell division phase. Yeah, and so they both encompass cell division. Even though mitosis and cytokinesis are slightly different. Okay, so that's just an overview. We are going to go through interphase and mitosis today, and then we're going to go through cytokinesis and some of the I guess some of the problems that can go wrong during cell division. Okay, so let's go into interphase. Now, interphase is the kind of, if you like, the normal function of the cell. Yeah, this is before the cell reproduces, and so it is kind of living out its normal phase. It's called interphase because it's between the cell division phases. Okay. This is when it does regular duties, it grows, it develops, and it replicates its DNA. Okay, so in a mammalian organism, the cell cycle is roughly on average about 24 hours. That is, it will take 24 hours from one cell to divide into two different cells, and then 24 hours again before each of those cells divide again. So it's a particularly slow process. Bacteria, for example, will take about 20 minutes or so to divide. So you can see there, 24 hours compared to 20 minutes is slightly different. Okay, of the 24 hours, the majority of it, 90% of it, or about 22 hours, is actually spent in interphase. So you can sort of see there, cell division is not a big part of the cell life. The majority of it is spent doing its regular duties, and then in the last kind of section, it does the cell division. Okay. It's marked by three particular phases. The first one is called the gap 1 phase. Yeah, the G1 or gap 1 phase is the regular function and the regular growth of the cell. This is when the cell grows in size, and then it also produces the necessary proteins because later on it's going to need to replicate its DNA to prepare for cell division. It also then needs to replicate, it needs to produce the necessary proteins for the mitochondria and the chloroplast in plants to replicate as well. Now, if the cell doesn't intend to divide, there are some cells that will not divide. For example, mature brain cells won't divide, in which case, at the end of this G1 phase, the cell, if it doesn't plan on dividing, will enter into what we call a G0 phase, which is otherwise known as quiescence. This means it sort of enters into this perpetual state of normal function. Yeah, it'll kind of carry out its duties for the rest of its life cycle, and it doesn't intend to divide, which means it's not going to replicate its DNA, it's not going to produce the necessary proteins for mitochondria and chloroplast to replicate because it doesn't need to. It's just going to simply continue on its regular function, and so forth. Okay. After it sort of grows, performs its regular function, and has all the important parts for DNA to replicate, it enters into the synthesis phase. And in this phase here, the genetic material of the cell is going to replicate. And so the cell increases its genetic material, and it doubles the number of DNA that it actually has in its nucleus. After that, it then prepares to divide. So then, now this is the part where the cell then begins and gets ready to split. So it grows a little bit bigger, and then it prepares to divide and split up all of its organelles and its nucleus into two different daughter cells. Yeah, and so those are the three main stages that interphase sort of marks. And then when the cell is sort of ready to divide, it has its DNA replicated, it has its mitochondria, and perhaps for plants the chloroplast, all the proteins and stuff necessary for replication. It then enters into mitosis. Okay, now mitosis can sound complicated at first, and it does take a bit of time to get your head around, but it's a particularly important phase in the stages of the cell life, and once you understand it, tends to be a fairly straightforward process. Okay, so definition of mitosis: it is the replication of genetic material in the cell. Yeah, and so basically it is when the nucleus of the cell divides. Yeah, now that's really important because it's marked by the nucleus dividing. Yeah. it's duplicating that nucleus that contains the DNA. So one of the key things to note is that the DNA is already doubled in number during the S phase of interphase. Now during mitosis, it's simply split into 2 different nuclei, each of which will go into a daughter cell. Okay, and it's marked by 4 particular stages, and each of them shows a different kind of action that happens inside of the nucleus. Okay, the first one's called prophase. The word pro meaning before. The second stage is called metaphase, meta meaning middle. The third phase is called the anaphase, ana meaning after, and then telophase meaning last. Yeah, so the words kind of give you an indication of where it happens. Yeah, pro before, meta middle, ana after, and then telo last. Now, before we actually start talking about each of these stages, it's important to understand how chromosomes sort of work and how DNA sort of is stored in the nucleus of a cell. Okay, so the DNA strand is coiled together to form what we call a chromatin. Yeah, it is a thread of DNA and all of the kind of associated proteins in that. So you've got your DNA in your double helix, and what it does is it coils together into these bundles around a protein called chromatins. And so these little kind of green dots here represent the chromatins. And then what happens is each of the chromatin then coil around themselves again, which then forms a chromatid. Now the chromatid is a tightly coiled DNA which becomes visible. Okay, when the chromatins become visible, we call it a chromatid, and this happens at prophase, which I'll discuss a little bit more later on. Once the chromatids join together into this kind of X shape that you typically see of a chromosome, that is what we call the chromosome, okay? Which is 2 identical chromatids that are joined together at this middle part here that we call the centromere. Here is a kind of a bit more realistic diagram of your chromosome. And so you can sort of see there, each of these things here is called a chromatid. The chromosome is the whole double strand that is joined together at this middle band here called the centromere. Yeah, and that's what it looks like under a micrograph. Okay. Now, how these chromosomes move sort of shows you which phase the cell is at during mitosis. Now, the first phase is called the prophase. Okay, the prophase is marked by chromosomes becoming visible as the pairs. Okay, as those X's that you typically see. So in the nucleus, the chromatin will shorten and condense to form these chromatids, and once they form the chromatids, it becomes visible. Before mitosis begins, if you look inside the nucleus, you won't see the DNA because it is uncoiled, and when it is uncoiled, it is too thin to be seen. But when it coils together, it condenses and it forms these chromatids, which then become visible. Yeah. So the chromatids then coil again and join together to form those chromosomes that you see. Now, during the same time, in the cytosol that is outside of the nucleus in the rest of the cell, you get these particular organelles called centrioles, which then move to the edge of the cell, and they begin to form this microtubules, which is a fiber-like network that we call a spindle. So what you're seeing here is that the cell is beginning to get its DNA coiled together and ready to split and divide, and then the cell is also preparing a network which allows it to move into its particular parts. The nuclear membrane, that is the actual outer casing of the nucleus, begins to dissolve. And so if I've also showed this in diagram, here is your cell, and then here is the nucleus, and the nucleolus is right here. So what happens is during the prophase, the chromatins will condense, and they coil together to form your chromatids. Now, the chromatids will then condense again to form these X's, and during this time, the nuclear membrane dissolves so that there is now nothing separating the DNA from the rest of the cell. Okay, these X's then attach themselves to this network of spindle fibers. These little peanut-like things are called centrioles, and your cell will have two of them, and they're going to move to the different sides or the opposite sides of the cell. You can sort of see there, one moving to the kind of northern side, and then the other one kind of moving to the southern side. And it moves to these different poles because that's the direction that the chromosomes are going to split into. Yeah, so these dotted lines represent this spindle network, and it's these fibers that are going to break those chromosomes into their respective halves. Okay, so that's the prophase. By the end of prophase and transitioning into early metaphase, what you're seeing is that the nucleus is not around. The chromatids have condensed into chromatids and become visible, and the centrioles have moved to their opposite sides. So that's what you're seeing, this kind of image here. Okay, when the cell then enters into metaphase, the chromosomes, which are now visible, will line up at the equator, which is the middle place, the midsection of the cell, and they attach to the spindle fibers. Yeah, these chromosomes actually lie horizontal because they're getting ready to split in half, so that each chromatid faces away from each other and facing towards the centrioles, which are lying at the poles. Yeah, the nucleus is fully dissolved by this stage, so there's no nuclear membrane separating the chromosomes from the rest of the contents of the nucleus from the rest of the cell. And that's what it looks like. Yeah, you've got this kind of lined up, every chromosome is lined up here along the edge. Now, one of the key things to note is that this diagram only shows 4 chromosomes. Okay, in a typical human cell, you have 46 chromosomes, okay, or 23 pairs, which will line up along the equator. And so this diagram only shows 4 for simplicity's sake, but there are actually 46 lined up right along here. Okay, and they're attached to the spindle fibers. And notice how each of these X's are lined horizontally, because this strand here is going to break apart and move this way up towards this centriole, and then this strand here is going to break away and move towards this centriole, but it hasn't happened yet. Okay, here is a microscope slide of metaphase in action. So you can still see there is your cell in metaphase at the moment. The nucleus is gone, so you notice how you can't actually distinguish which part of the cell is the nucleus because the membrane has dissolved. You can see there these little fibers that are there. The centriole would be right in the middle here. You can't see the centriole, most likely because it's too small to be seen, but you can see the nature of the fibers forming this kind of network, and these network lining up your chromosomes along the middle. Yeah, and you can sort of see these strands here and there showing the chromosomes lined up along the middle. So you can tell there that if the cell splits, it'll split right down along the middle. Half the contents going to this side on the left, the other half going to this side on the right. Yeah, and so that is a snapshot of what metaphase looks like. Okay, it hasn't split yet. Yeah, and which is what defines metaphase, that it's lined up but it hasn't split. Okay, now when it does split, that's when it begins to enter into anaphase. Yeah, so anaphase is the third stage. The centromere divides, that is, that little spot in the middle of the X on the chromosome divides, and each of those single-stranded copies of the chromosomes, the chromatids, will move to the opposite ends of the pole. Yeah, each chromatid will move towards the poles. The spindle fibers will shorten, that'll pull the chromatids away from each other, and then each half of the cell will now have identical DNA to the other half. Okay, so there you've got that action. The centriole here will pull the spindle fibers towards itself, and that pulling action will then break and split the centromere, and then each chromatid will be split. Yeah, so this chromatid here will have almost the exact same or identical DNA as this one here. Yeah, and so that one and that one will be identical, that one and that one will be identical. The only exception would be if there was some sort of mutation or something that occurs during this phase to have changed the DNA, but otherwise we can expect this particular stage to be completely identical on this half here to this half here. Yeah, otherwise the process wouldn't produce identical daughter cells and we would be slightly in trouble. Okay. There is another microscope slide of anaphase happening. You can see there now that darkened patch there is the chromatids, which have now broken apart, and that middle section has been divided, and the cell has already started to show signs of that kind of division happening. Okay, so there's anaphase, and that's the snapshot of what it looks like. Okay, now the last one is telophase. Now in telophase, the cell then needs to start reforming itself into a proper cell. So the two sets of chromosomes must form tight clusters again. The chromatids have all joined together, and so they cluster up again. A new nucleus needs to form, so the nuclear envelope needs to reform around the DNA of each daughter cell, and so the cell will now have two nuclei instead of one. Yeah. Now you got to remember the cell hasn't fully divided in half yet. Only the nucleus has divided in half to form your two nuclei, and so that's why you have two nuclei forming in the one cell. The spindle fibers then they've done their job, and so they disassemble and they begin to disappear back into the centriole. Yeah, the chromatids will then uncoil back into the chromatins, and because they uncoil, they're going to become less visible, and so the cell is now has two nuclei on each side of, or on each pole of the cell. The centriole has shortened the spindle fibers, and the nuclear envelope has reformed around each cluster of chromatins. Here is a microscope slide of what it looks like. You can see there the cells already begin to divide, but it hasn't fully divided yet, which is what the next stage is at. But you can see there it's clustered up again. The spindle fibers have done their job, and it's basically ready to become two new cells. And that is a snapshot of telophase. You typically see the cell already beginning to kind of warp into its two daughter cell shape, but it hasn't fully divided yet. Okay, so let's do an overview, and then in the next video we're going to look at how the cell actually then divides the rest of its contents into two daughter cells. But this is just interphase there, and as the cell enters into mitosis, you've got four different phases. Prophase, and during prophase, you've got the centrioles forming and then moving to their opposite poles, which is what forms the network that will attach for the chromosomes. The chromatins, which were invisible previously, will condense and coil up to form the chromatids, which now become visible. The nuclear envelope, that is the membrane that surrounds the nucleus, dissolves, and the chromatids will then begin to form your X's, or the chromosomes, and then they start attaching to the spindle fibers. Okay. By the transitioning stage into metaphase, this is the snapshot that you kind of get. Okay. The chromosomes then line up at the middle during metaphase, and they lie horizontal in the equator, and they attach themselves to the spindle fibers around this particular stage here, getting ready to split in half. In anaphase, the spindle fibers then shorten, and it pulls the centromere apart of each chromosome, splitting them half-half, half of the identical DNA into one end, half of the identical DNA into the other end. Okay. By telophase, the spindle fibers have done their job and have begun to disassemble back into the centriole. The new clusters of chromatids will begin to uncoil into chromatins, and a new nucleus is going to form, resulting in two nuclei forming on opposite sides of the cell. The cell begins to take the shape of a cell that is about to divide into two. Okay, and then later on, when we look at cytokinesis, the cell would then divide the rest of its body into two cells, which are now identical to each other as daughter cells. Okay, so that's just an overview summary of mitosis. It's important to understand each of these steps, because later on when we sort of investigate sort of how these steps play an important role in determining the genetics of an organism, this chapter will sort of set the foundation for all the other chapters that you guys are going to learn. Okay, so I would take the time to make sure that you guys are aware of the processes, the ins and outs of each stage, and you are well aware of this particular summary page here. Okay, alright, so that's it. I hope you guys enjoyed that video, and next time we're going to look at how the rest of the cell divides and some of the issues around the cell cycle. Okay, see ya.